My name is Timothy Tardivono. I'm the Vice President of Public Policy for Connect. Thank you all for coming out this afternoon. We were over on the House side this morning, and we wanted to make sure the Senate had a chance to come meet Dr. Reed and Mr. Salka. So Connect is you could call us an innovation accelerator. We help tech startup companies grow. We're based out of San Diego, and we've been doing this for about 25 years. And about 25 years ago, the leaders of San Diego saw that there was a lot of research, a lot of scientific activity, but it wasn't turning into companies and jobs. And they got together and they said, well, how do we make this better? And so they created Connect as part of the University of California, San Diego, to help with the development of those companies. So we help startup companies sort of go through the process. We mentor them, we get them connected to potential funders, to other people in the industry that are moving along. And so over the last 25 years, we've helped to start over 2,000 startup companies. It's sometimes a little hard to track all of those because we consider a company that's merged or acquired as a successful exit. So they're not all still in existence, but we continue to do that. We have a program called Springboard that's an intensive sort of 6 to 9 month mentoring program that helps these startup companies walk through that process. It's hard enough with the market forces, but we help them to not make mistakes that could be fatal to their company. So we started there at UC San Diego. We are fortunate in San Diego to have a thriving research sector that is putting out great research and really starting to has been propelling commercialization. Today we're featuring Sanford Burnham, but we've got everything from the Scripps Institute itself. UC San Diego is a leading research institute. The Von Liebig Center is UC San Diego's accelerator that helps academic research advance. And then, of course, we have SPAWAR, which is the Naval Research Lab that does a majority of the research not all over the country, but specifically West Coast related things. They're one of the leaders in the cybersecurity efforts that the Defense Department is doing. We are fortunate in San Diego that we have multiple clusters. Some cities and regions have emphasis in certain areas. San Diego is broad-based in the kinds of new technologies. We started because of the presence of the military and the Navy years and years ago in the 40s, and now we have over 300 defense and security companies. Wireless and software. Qualcomm is a story that started as an SBIR grantee and is now a major telecom and chip maker that is in most of your phones that you have. Qualcomm products are in there. You can see some of the others. And of course the life sciences companies in the late 70s and 80s. Some people argue that the life sciences industry was birthed in San Diego because of the research that was done. And here you have our company we're featuring today, startup company Sendar, and he'll be getting into the real novel approaches they're doing with cancer that can really make a difference. Over 600 life sciences company in San Diego. Because of our great weather and our exposure to the sun about 350 days a year, we're a leading solar energy producer, and we are also are seeing that these different sectors are beginning to converge. So our alternative energy and our life sciences clusters are merging to now we're leading in the biofuels sector, algae biofuels, others. The Navy is starting to commission their first ships fully run on biofuels, and it's right there in San Diego. And our funnest probably is our sports and action industry. Obviously everyone knows you got a lot of surfers, skaters, bikers, mountain climbers, everything's in San Diego. There's so much to do outside. And we are developing a new, what we call San Diego Sports Innovators, headed by Bill Walton, the famous NBA player who's from San Diego, UCLA. But he is finding all these guys in San Diego that can make a better surfboard, a better skateboard, a better shoe, a better shirt, a better helmet, and they're realizing not only can I have fun, I can actually start a company and compete against the Nikes and those others. And so that's Connect focuses on helping other trade associations in specific areas to move forward. We sort of are more of an umbrella organization that is sort of agnostic to what kind of technology you are. If you come to us and we think you're viable, we will help you. Last year we won the Economic Development Association Administration's Innovation Award for Regional Innovation Clusters. And as Congress is looking at what kind of jobs do we need to get this recovery going, we really would impress upon you to think about how can we make the tech sector grow. And the reason is because of what we found in San Diego, which is that the number of employers, only about 6% of the employers make out about 11% of all jobs, 25% of the wages, but those wages are 114% higher than the average industry wage in the non-tech sector. And so you can see here the price for a non-tech sector wage is about $46,000. In the tech sector, the average wage is around $97,000. And here you can see all the different kinds. Each of these represent a different sector. And so these jobs are highly skilled, but they're high paying. They bring 2 to 3 jobs with them in other support, either research or administrative. And so when Congress is looking at how do we really get the economy moving, we would argue that the tech sector is the place they should consider. How do we incentivize tech sector employment and allow these tech sector jobs like we're going to hear about today to grow? And so without further ado, I'll bring up Dr. Reed. He is the CEO of Sanford Burnham Research Institute. They have a very difficult time recruiting employees because their bases are Santa Barbara, San Diego, and Orlando. So it's real difficult for them to convince people to come. But they're bi-coastal, they're in Florida and California, and they're one of the top 3 research institutes in the country, especially related to NIH funding. And Dr. Reed's going to talk a little bit about the impact of NIH funding today. So today, welcome Dr. Reed.
Well, thank you all for taking time from your busy schedules to join us for our program and for us to have the opportunity to share with you a little bit about this important ecosystem that exists in our economy, where basic research in the life science and medical research area creates the innovations, the intellectual property that result in patents that help to spawn new companies and create jobs in this country and help us to be more competitive on a global level in the 21st century, where it's said that biotechnology will be to the 21st century what information technology was in the last century. What I'm going to do is focus most of my remarks today on helping you understand the catalytic role that NIH funding, the National Institutes of Health, plays in supporting this ecosystem of economic development. And I'm going to touch on about 4 different areas. Before I start, though, I want to just show this slide, and it says if you don't get anything else from the talk, it's this, and that is that the investment our country is making in medical research through NIH is not only paying dividends in terms of improving healthcare outcomes for our country, but also is helping to lay a foundation for vibrant economic growth. So the things I want to touch on are illustrated here. So first I want to talk about the way that NIH-funded research in this country provides the support that is a whole platform for an industry that generates revenues with then taxable revenue streams back to the federal government that makes this sustainable several times over the investment that the federal government makes. The second I want to talk about is the competitiveness at a global level with respect to the innovations that come out of this research and our ability to capture intellectual property and use that as a base of creating new jobs in this country and securing the future for our country in an increasingly competitive era. I won't talk about today, but it's also important to note that NIH funding of medical research also plays a big role in training the next generation of scientists. And the current challenges with the NIH budget, where it's never been more competitive in the history of the NIH than it is today with respect to the percentage of grants that the NIH can fund, is creating what has been referred to by the National Academy of Sciences as a broken pipeline, where our best and brightest scientists are unable to stay the course, and we're beginning to see a reduction in the numbers of PhDs who are coming out of the pipeline and able to then staff our labs. In fact, in 2010, China surpassed the United States in the number of PhDs, and so we no longer have the most PhDs in this country. It's also leading to a brain drain, where some of our best scientists are leaving this country and seeking opportunities in other nations where they're ramping up support of medical research. So that's all I'll say about it today, but it's a very important aspect. And finally, I want to talk about the repercussions of this investment we make with respect to healthcare costs. We're all concerned about what it costs to provide healthcare to our nation. We believe that through these innovations, we can see new opportunities to make the practice of healthcare more efficient and drive down costs. So first, in terms of understanding this ecosystem, what I want to share with you is that the way that new medicines and new medical products come about is illustrated in this slide here. It starts with basic research, where we understand what causes disease. From that, we identify what we call the targets for drug discovery, where we now know what we want to fix. We then generate prototype medicines, often referred to as lead compounds, which then begin on a path, complicated path through clinical trials, phase 1, phase 2, phase 3 trials, and if all goes well, FDA approves, and now a product comes out that can help to improve the lives of patients as well as to create new economic opportunities. Now, the funding for this starts largely with NIH. The NIH funds about 80% of this sort of activity in our country, and that's about a $31 billion investment that creates around 350,000 jobs. The important thing to note about this, though, is that most of those dollars are not spent here in Washington or in the government. Most of them, 85%, are distributed around the country to research institutes, research hospitals, universities, medical schools that do the work. So only a small percentage of it is actually done in Bethesda, where the NIH campus is. But that basic work there is the underpinnings then for the rest of the pharmaceutical industry, because with those discoveries then, these new product opportunities can then move into the hands of the biotech and biopharma industry, which in this country alone spends about $70 billion on R&D, which creates another roughly 600,000 jobs. In addition to that, there's also the medical device and the medical laboratory industries, which are also spawned in large part out of this, which creates another almost 900,000 jobs. So it's safely estimated that every dollar that we spend in the NIH generates at least $3.5 additional dollars in economic activity in our country. Now, you can make pretty decent arguments that that investment also comes back in the way of tax revenues to more than pay the government back for the investment we make. If you look just at wages and income tax on wages, if you look at the jobs funded by NIH grants and the average wage, and you look at jobs in the life sciences industry and the average wage, those two together generate about $60 billion in income taxes that come back and more than twice repay the federal government's investment. And that's not even to account for the corporate tax opportunities that arise from profits of for-profit companies that are in this space. So that gives you a little sense of the sort of the feeding chain that exists here, from basic research through to products that help patients and to create economic development. The other thing I want to touch on next is the role of NIH funding in terms of creating the intellectual property, the inventions, the innovations that allow our country to be successful. It's firmly believed that America is really a birthplace of innovation. It is clearly what we have as our greatest strength on a competitive basis, our ability to innovate. We can't necessarily out-manufacture the rest of the world anymore, but we can out-innovate, and we've shown we can do that. It's estimated that about half of all the world's great inventions have arisen from this country. I think all we have to do is reach in our pocket and pull out one of our favorite devices, and we can look at the innovations that come out of this country, the industries we spawned. So what is and the other important thing about that is if you look at balances of trade, and you look at various sectors of industries, one of the few areas where we still have a robust surplus rather than a deficit in trade balances is in the area of intellectual property, and that is how much revenue do we obtain as a nation for others to have access to our inventions, paying royalties, licensing fees, etc. And in fact, we lead the world by far in that. The closest to us is Japan, which is maybe 1/10 what we are. So it is clearly an area where we excel. And this has not been lost on the rest of the world. This is a quote from the Premier of China. It says that the future world of competition will be for intellectual property rights. So here too, the NIH funding of medical research plays an important role, because the investment that's made in funding extramural research by NIH, about $25 billion coming into the academic nonprofit world, generates intellectual property, which then typically is licensed by small biotech companies, which are nurtured in part by small business innovative research grants that come from NIH, but also by venture capital. So we attract an equal amount of capital into our economy, roughly $30 billion a year from VCs. And then finally, these products are often then acquired by large pharma companies, which invest another $70 billion. So you see that the investments made here catalyze further downstream investment and then lead to products which can create a revenue base through taxes that help to keep this a sustainable system. NIH grant recipients are actually obligated to file patents for what they invent. This is thanks to the Bayh-Dole Act, which made that an obligation of those of us who receive these. Interestingly, 74% of all U.S. biotechnology and pharmaceutical companies have licensed NIH patents. Over half of them, 59%, say that those patents are fundamental to the strategy of their company. Nearly 20%, or 1/5 of all products eventually approved by FDA, can be linked directly to NIH-funded research. I think some interesting statistics is if you look at NIH grants, there are roughly 40,000 of those that are currently funded across the country. 98% of them result in an invention disclosure. Quite an impressive number, I think. 56% result in a patent application, and 22%, nearly 1/4, result in an issued U.S. patent. So this is an illustration of how this funding creates innovation that leads to patents and allows us to stake out territory from its intellectual property standpoint that help to create jobs in America. The third thing I want to talk about is healthcare costs and what we can do about them from the standpoint of innovation. We're all concerned about this. We've all seen the numbers in terms of the rate at which healthcare costs are rising. Of the 10 leading developed countries of the world, we spend the most on healthcare, 2 to 3 times per capita more than other nations, and yet we rank 9th or 10th in all major categories of healthcare outcomes. We firmly believe that the way to address that is through innovation. Already, through NIH-funded research, a number of innovations can be pointed to that have huge savings. I just list a couple examples here. At the top is the drug for Alzheimer's that was discovered by one of our researchers at Sanford Burnham, called Namenda or memetine. This is a medicine that prevents, slows the rate at which brain cells die in patients with Alzheimer's. And clinical trials have estimated that it is able to forestall the time at which patients with Alzheimer's become dependent and have to go on to assisted living by about 18 months on average. That's estimated to save the nation about $100 billion annually in costs related to caring for patients with Alzheimer's. So we spend less than $1 billion as a country to provide that medicine. We save $100 billion in the costs that occur as a downstream of that. Another example would be the HIV medicines. When I was a medical student, the hospital wards had many patients with HIV infection, horrendous hospital courses of months at a time, very expensive. Now, nobody's in the hospital for HIV. We have drugs that take care of it. You rarely see it. So these are just a few examples, and I could go on and on. But even more exciting, I think, is the juncture at which we are now, thanks to genomics and some of the wonderful developments in technology that are beginning to make it possible to realize an era of personalized medicine where we can begin to transform the practice of medicine from less of an art and more into a science. Underlying this is the technologies related to next-gen DNA sequencing. The ability to drive down the cost of DNA sequencing is absolutely phenomenal. About a decade ago, it's just a decade ago that we celebrated the sequencing of the first human genome. It was a project that took 13 years, involved thousands of scientists, and cost about $3 billion. 2 weeks ago, a US-based company announced an instrument that will sequence an entire human genome in 2 hours for about $1,000. This is going to transform healthcare, and this country needs to be at the forefront of that. What kind of things can we imagine, and how might they impact healthcare? Well, let's take just one example of how this type of technology could save money in our healthcare system. I look at an example here of heart failure. There's about 1.5 million Americans that have heart failure. And many times as these patients end up in the hospital where they're not properly receiving the right medicines, etc. Medicare costs alone in this country are $17 billion a year for what are referred to as readmissions for heart failure, where a patient came in, was hospitalized, was treated with various medicines, looked like they were better, sent home, and then they were right back again where we hadn't really gotten it right. So one of the studies to illustrate some of the things that could be possible in the future is a study done here by one of our scientists, Dan Kelly, identified a genetic difference in a gene that's involved in metabolism in the heart, where some of us are born with a G in this position and others an A. And they looked at the rate at which patients are re-hospitalized after admission. So if you have Gs at that position, where both your mother and your father gave you a G at that position, your rate of hospitalization when you come in, re-hospitalization when you come in with heart failure is very high. But if we treat you with beta blocker therapy, it goes very low, down to this dashed green line. On the other hand, if you are born with A's at this site, then you have a very low rate of re-hospitalization initially, but if we give you the same treatment, what's called beta blocker therapy, you actually have a higher rate, so you've actually done harm to the patient. Prior to knowing this sort of thing, you can see the inefficiency in the healthcare system, right? So this is the kind of thing that we can begin to address. Now, genomics won't take care of all this. It's clear that not only the genes we're born with, but also the environment in which we live and operate is playing a big role. And we're all very aware of this in context of things like the national epidemic on obesity, where about 30% of us now meet clinical criteria for obesity. So to be able to look in a more integrated way at what's going on, there are other technologies, metabolomics, proteomics, together with genomics, that we can begin to use to get a better picture of what's going on in the patient. And I'll illustrate just one example of a proteomics technology from my laboratory to illustrate this, again, in the context of how we might improve healthcare efficiency and therefore drive down costs. So I'll do this in the context of prostate cancer. All of us men, at least in the audience, are very concerned about prostate cancer. Prostate cancer is essentially a disease of aging. If you live long enough, you'll get it. Autopsy studies show if you're 75 years or older, 85% of us men will have prostate cancer whether we know it or not. Now, the conundrum is that actually for most men with prostate cancer, it's a very indolent disease that frankly doesn't need to be treated. You're more likely to die with prostate cancer than of prostate cancer. But for other men, it's aggressive and needs to be treated. How do we know what to do? This is a great dilemma. But some of these new technologies are beginning to allow us to find the molecular signatures in patients' tumors that can tell us whether it would be sufficient just to watch and wait and monitor, or whether aggressive therapy is needed. I'll show you just one example of this where we identified a protein called BAG1 that binds the androgen receptor and monitor the outcome of men after prostatectomy. There's a couple different ways of looking at the data, but what we're looking at is the number of patients who, where the disease relapses over time. So start off with 100% relapse-free, and then it reduces over time. If your tumor has this BAG1, then the relapse rates are very high, so very few patients remain disease-free at 5 or 6 years out. Whereas if it's negative for this, then most of those patients, 80%, remain disease-free. Again, not a perfect solution, but it begins to give you an understanding of how we can start to layer this kind of technology into healthcare practice and make more informed, more intelligent decisions that will help to drive down costs. Another exciting vista of opportunity for us is thinking beyond simply analyzing molecules in cells, the DNA, the proteins, the metabolites, and to being able to develop personalized medicine around cellular functions. Here there's some wonderful advances in stem cell technologies that are beginning to create a whole vista of opportunities for us. One version of this is illustrated here, and I've done this in the context of thinking about neuropsychiatric diseases. Why neuropsychiatric diseases? This is an area where there's a great deal of inefficiency in healthcare. The average patient who presents with depression, bipolar disorder, anxiety, schizophrenia, has to go through 5 different medicines in a trial and error basis before they find one that might work for him or her. So it's an area where there's a great deal of inefficiency. Very common disorders. 1 out of every 3 of us at some point in our life will have a major mental health disorder. 1 out of every 6 Americans right now has some version of a mental health disorder. And it's the largest selling sector of pharmaceuticals among all therapeutic areas. So some of these technologies now allow us to, for example, take a cell from a patient, typically it's a skin cell, synthetically, through putting a cocktail of 4 genes into those cells, to turn them into stem cells. And then once made into stem cells, we can program in the laboratory to produce, for example, nerve cells if we want to study a neurological disease. The importance of this is that previously you couldn't get people's brain cells to study them. It's not very likely that you're going to do neurosurgery on the patient and take out a few brain cells to find out what's going wrong. But here we can make them synthetically in the laboratory. And so the idea then is to be able to then take those brain cells, which we've created from the patient, test in the laboratory, in the Petri dish, whether they respond to medications, and then to use that information to then inform the physician, the patient, what medicines to take. This idea actually has been spun out into a company from the Institute called NeuroMap, where they're attempting to now develop this at a level where it could provide a clinical service. It actually won an award for entrepreneurism as the best idea that came out of a whole competition. And it illustrates just one example of some of the types of opportunities that are beginning to emerge. The other thing that becomes possible from these, what we call disease in a dish, disease in a Petri dish, sorts of opportunities where we can take cells from a patient, recreate the disease in the Petri dish, and screen for responses to medicines, is that we can also begin to not just think about what medicines are approved for that disease, but to think more broadly, if I could take all the medicines in the world that are already approved and try them, is it possible that just through serendipity that some of these medicines might work for the patient? And we're already seeing examples of that in some of the things that we're doing, for example, with children with muscular dystrophies, where we can do the same kind of thing, make muscle cells in the dish, screen the collection of all FDA-approved medicines, and finding medicines that we never would have expected might help muscular dystrophy, fixing the problem in the Petri dish, and now we're testing whether it will fix it in the patient. So I think some enormous opportunities. Already we can estimate that the NIH-funded research has generated, conservatively speaking, about, since 1995 alone, about $70 billion in annual savings in various medical costs through a study that was published and others. We can also even think, if we go beyond direct medical costs and think about all the other indirect costs of illness, there's a pretty good argument to be made that this investment that the nation makes in funding NIH research, which really provides the underpinnings of this whole industry, is more than paying for itself. In fact, by one estimate, it's estimated that that the annual savings of all costs is about $3 trillion annually, which is a 100-fold return on investment relative to what we invest as a nation. So I will then conclude my remarks here, and we'll introduce Dr. Scott Halka, who is our next presenter. One of the things I want to emphasize is that from the standpoint of somebody who's involved in a nonprofit organization running a medical research institute, is that while we do the discovery research, the sorts of things that go on in medical schools, research hospitals, etc., we don't actually manufacture anything, we don't sell anything, and we require partnership with the for-profit companies of the world to see that this wonderful research gets translated into products that can help patients and help grow the economy. Scott is a wonderful example of the kind of entrepreneurism that exists in this nation. He's been involved in starting biotech companies now for 20 years. He's now on his sixth company, a serial entrepreneur, and he's going to tell you about some technology that his new company is developing that was licensed from the Sanford Burnham Medical Research Institute. Scott?
You know, I know I'm doing exactly the right thing professionally when I can sit and listen to this for the second time today and get really excited about the work going on at Burnham. It's just really cool stuff. And I'm not a doctor. I play one on TV. I'm a finance guy that got involved in biotech 20 years ago, and it has been one heck of a ride. So I'm going to talk a little bit about, as Dr. Reed was mentioning, a little bit about what I'm doing, my new company, and how we are taking some really cool technology out of the Burnham. And then I'm going to wrap up by kind of drawing a thread through some of my experiences and what Dr. Reed was talking about. So real briefly, oh, and I wanted to mention too that, I don't know if it's indicative of a hierarchy or if it was just happenstance, but at the house this morning, we were in a subterranean room. I think the floor was tile, and they served like saltine crackers and cane. And then you come here, and it's really nice here in the Senate. So you guys have great digs. So as Dr. Reed mentioned, this is my 6th company, and it takes a long time to develop a drug. Dr. Reed was talking about a long time that technologies have to incubate inside of academia before they can be commercialized. But even once you take some insight that's developed in academia and you go down that path of getting a drug developed, it takes a long time. My last company has a drug that's now in phase 3 clinical studies. Best case scenario, and this is for a cancer that currently is not well treated, that if you get diagnosed with this cancer, you'll likely be dead within 12 months of diagnosis. Nonetheless, it'll likely be, at best case scenario, another 3 years before it hits the market. And the phase 1 filing was in 2006. So that's just an example. And then there was a hedgehog inhibitor that Roche just got approved. You might have seen it in the news for basal cell carcinoma. And they talked about how fast, FDA was crowing about how fast that got approved. And it really did. It was an accelerated approval process that the FDA should be given kudos for putting in place. But if you look at the real story, that drug, the IND for that drug was filed in 2006. So fast is fast, right? So one of the things I wanted to do for my next company was do something that I had a chance at getting into the clinic and through the clinic before I turned 60, and I just turned 50. So so the sender technology kind of fit that bill perfectly, because it's not a drug. We were talking this morning, it's kind of like those old BASF commercials, we make drugs better. It's a really interesting peptide that was discovered at the Burnham. Peptide is just this little fragment of a protein that when you inject it into a patient with a tumor, that tumor all of a sudden behaves very differently than a typical tumor that's trying to push everything out. This tumor all of a sudden starts bringing everything in. So we can co-administer it with the drug and get a lot more drug to the side of the tumor and make those drugs work a lot better. And it works exceedingly well, at least in preclinical tests with small molecules, with chemotherapeutic agents, with biologics, antibodies. So that's kind of what that's the technology that we took out of Burnham and that we're now commercializing. So again, if we can get more drug into the tumor, deep into the tumor, obviously increase efficacy. Very important to reduce side effects. Chemotherapies, as you know, other cancer agents, as you know, just really wreak havoc on the patient. So if we can lower the dose required, we can reduce cardiotoxicity, toxicities in the brain, your hair falling out, gut toxicities, but still maintain the efficacy against the tumor. So this really represents a truly, I think, a potentially groundbreaking technology right out of the Burnham. And I've got to say that this technology is based on some insights that were first hatched probably 25 years ago. So it takes a long time in academia for these types of insights and discoveries to mature and get ready to go into commercialization, early commercialization. So it's a nicely peer-vetted technology. We founded the company after some discussions with the discovering scientists in late 2010. We completed the license from the Burnham in late summer of 2011. And then we've been really focusing on getting grant funding to push this along, and I'll talk about why in the next slide. But we've been very fortunate because of the status of the science that we've been able to get NIH funding. It's special NIH funding like SBIRs, STARTs, that are really focused on that translation from academia to commercial entity. So we've got $2.5 million worth of grants that are funding everything that we need to do to start our first clinical trial with this peptide. I have a couple science slides. I'm just going to stick to this next one here. As I mentioned, it's this little peptide, this protein fragment, and what it does is very interesting. The mechanism by which this kind of pulls off, this trick it pulls off. It homes directly to a receptor that sits on the outside of cancer cells, and it's really highly upregulated in cancer cells. So the peptide actually moves to the outside of the cancer cell, and then when it's there, this event happens, this protease cleavage event happens, and it exposes this cryptic binding site that goes to a different receptor on the cancer cell and triggers this bulk transport mechanism where the tumor starts drawing everything into itself. So the combination of this receptor, this protease cleavage event, and then this other receptor makes it very, very selective for tumor cells. Because as you might imagine, our body didn't evolve special cancer receptors. These receptors are also used in normal tissues. And so this perfect triumvirate of events that need to coincide for this to work only happens on tumor cells. So it's very selective for making tumor cells or tumors leaky and not normal tissues. Here's a cool picture of this peptide working. This was published in Science. So our peptide is labeled with a green fluorescent dye, and this is a slice of a pancreatic tumor. And this is 15 minutes after we put the peptide in, 30 minutes after we put the peptide in. So it happens very, very rapidly. And you can see that at 15 minutes after administration, you get localization, starting to get localization of the peptide around the tumor. And then even 15 minutes later, you see invasion into these tumor ducts, where in the absence of this peptide, your agent would just kind of sit out here and kind of act against the tumor here, but not in some of the critical areas of the tumor. So it really allows us to quickly access areas of the tumor that are typically not accessible to drugs. Here's a great picture of our peptide with Herceptin. Herceptin is a HER2 antibody developed by Genentech, now Roche. These things are really expensive. To treat somebody with a biologic is, you're talking tens of thousands of dollars per year. So here we stain the tumor sample, and the Herceptin shows up in this rust color. And you can see this is 3 hours after administration. This is a breast cancer slice. And you see a little bit of Herceptin in the tumor, but that same tumor, this time with the Herceptin co-administered with our peptide, you see just a lot more drug into the tumor site. So from a cost-benefit standpoint, you get better activity, and you can use it on a much lower dose of the drug. So with an antibody, which antibodies aren't typically toxic in and of themselves, although some of these antibodies do have cardiotoxicities, but you can reduce by a significant factor, maybe use 60% less antibody, which has a big impact on health costs. But when you're talking chemotherapy agents, even though those aren't necessarily that expensive, you are sparing the patient's heart and liver and gut from some of the deleterious effects of chemotherapy. So here are some really important, it gets into the tumor, but does it work? Is the mouse, in this case, better off? So here's an example of anti-tumor activity in a mouse model. So we grow tumors up on a mouse. And here is an example of Herceptin alone. And we tune it so that Herceptin alone is at a high dose. The tumor is just kind of motoring. It doesn't grow, it doesn't necessarily shrink. And then that same dose of Herceptin with our peptide, and you can see the tumor just shrinks. So that's obviously makes us feel good about the prospects of this technology in the clinic. Now here's an example. This is still an on-patent drug. It is an old drug, a taxane, that is formulated with a human serum albumin, which makes it a much easier drug to administer, a much safer drug to administer. But it's also very expensive. So we tune this model so that A typical dose, this dosage of Abraxane alone does nothing, it does not behave better than vehicle. And then we combine IRGD, our peptide, with Abraxane, and you see a much better tumor response. So another example of how we can improve the efficacy and reduce toxic side effects. So the objectives that we have just for this year, and again, we're going to do all of this with grant funding, is to initiate our first clinical study in 2012. Now, one of the really important things, one of the things I like as a commercial developer, but also just from a patient perspective, because this works co-administered with drugs. We're not creating new drugs, so we don't have to go through the entire approval process. We're not creating a new chemical entity. It's essentially a helper, an adjuvant for drugs. So we just have to get a safety picture of the peptide by itself, a safety picture of potential interactions between the peptide and the drug, and then prove that it works. So it's not too long of a clinical study. We expect toward the end of 2012, after having completed some preclinical pancreatic studies, to start our first gemcitabine IRGD combinations in pancreatic cancer. And then, importantly, this year we expect to have our first partnerships with pharmaceutical companies, biotech companies, to help accelerate the commercialization. I have a slide here talking about going, the dilemma, the challenges, the fun of bringing science from the lab to the bedside, and the critical role of the NIH in funding that path. So I mentioned up front, I'm a finance guy, steeped in efficient market theory, and I'm in general an efficient market guy, but I think you can kind of toss out the notion of efficient markets when it comes to the kind of stuff that Dr. Reed is doing at the Burnham, and what we tried to do is we translate the science. When I started my first companies in the early 90s, venture capital was awash in the space. You could get a mouse that had some interesting effects, a mouse model of human disease, a couple guys like Dr. Reed at the Burnham, and I could go out and raise $20 million, and we'd be off to the races. Pharmaceutical companies had PE ratios that were more like Google's PE ratio, so money was just flowing into the space. Fast forward 20 years, it's not like that any longer, and because of the financial meltdown, it is the early stage venture capital has taken it on the chin. What venture capital does exist in our space is mostly focused on kind of very late stage clinical programs. Very little capital flows into preclinical work. So we really rely on the NIH to help fund this stuff. And some of the forward-thinking biotech, larger biotech and pharmaceutical companies who, once we use some of these early NIH funds, we can entice them into starting to fund the work that we do. So I want to encourage you guys to consider what Dr. Reed and I have said and help us keep this country at the forefront of technological innovation. Okay? That's it. Thank you very much.
We do have time for a few questions. If anyone has any questions, both Dr. Reed and Mr. Salker are happy to answer anything.
Getting back to this venture capital problem, so is there a government solution to approach to this? I know NIH has got INCAS that they're trying to address it, but how do you solve that problem?
I don't know that the government needs to run in and try to solve it, because I am an efficient market fan, and I think that in time this inefficiency will be kind of ironed out of the system. But for the time being, I think some of the things that the government can do is make sure that the NIH funds, some of the programs that are already in place are well funded, and so so that money can continue to flow into this gap right now that exists between the work that's done at institutes and the work that needs to be done before the market can kind of pick things up again, which is kind of later than where you guys drop things off right now.
I guess one of the vehicles of that is the Small Business Innovative Research Grants, the SBIRs, which is about $2 billion of the $31 billion the United States spends every year is devoted to for-profit companies to help nurture the technologies along a little further, get them to certain inflections in their milestones that maybe now a venture investor or a biotech, biopharma company would pick them up. So I think that's that's an area that could be expanded more.
And I think they are expanding it, but it's expanding to levels that it existed as a percentage at 10 years ago. So there is an expansion, but I think if there's a way that government can figure out how to maybe even goose it up a little bit more over the next couple of years.
And it's actually not new spending. The SBIR percentage comes out of the total research budget, and the bill that just passed increases that from 2.5 to 3.2%, but it's over a period of time. So that increase is gradual and slow, or maybe you all would like it to be a little more protracted a little bit.
If you look at the percentage of the NIH budget that goes to kind of translational research, it is still relatively small.
Mm-hmm.
Right here.
Yeah, very fascinating. The efficient market theory is one that we can debate, but assuming that we move forward and there's patents secured and eventual payout for those patents as this is commercialized, and there's licensing fees that will help pay along the way. And then ultimately, the technology is going to generate more dollars, more jobs, and more taxes for our economy. But is there a direct payback to the NIH for those investments that they're making at this early stage?
There's not. So, except, maybe one could argue that the Burnham, for example, owns a pretty significant chunk of Sender at this point. And if we are successful at funding the company with these grants, which are non-dilutive, so NIH gets no stock ownership in the company, then Burnham will end up owning much more of the company than they otherwise would have, and then a lot of money can potentially flow to Burnham.
Right. His organization can do further research.
Exactly.
That's right. So again, about 85% of the NIH budget is spent extramurally, about 15% here in Bethesda, but about 85%. And the government does have, through the buy-dole provision, that we're obligated to file patents on intellectual property and to license those with preference to American companies, particularly small companies. But then the hope is then there'll be revenues that are returned to the inventors, the institutions that hold those inventions, that we can then put back into the research enterprise and help to further promote more research.
Question right here.
You're talking about a 6-year, very long preclinical trial period. So you've been in a good scenario, and I understood you guys correctly, it was about 6 years or some long number
That's
of years.
very, very fast. Yeah.
Is there any time in the foreseeable future that it could go down to half that, or is there anything that the government is doing or could do to shrink that period?
I think there's a lot that can be done,
And then maybe a follow-up to what haven't they done here?
they are working on it, and there have been examples. So imatinib, which is Gleevec, it's a Novartis drug to treat CML. It went from first dose in man to approval in 3 years. So there certainly have been times in the past when that's happened. I don't think that would happen now under today's leadership at FDA. And a lot of that is, I think, just the risk aversion that's been baked into that organization prevents them from taking some of the risks that I think they perceive they're taking by giving accelerated approval.
Yeah. One of the challenges, I think, or the frustrations, I think, for developing our economy is that while the pharma industry spends $70 billion in R&D in our country, they spend actually more than that externally because of the frustrations over working through the FDA process. So it's most likely that the typical route is for a company to first try to get their product approved in Europe because it's a system that works in a more cooperative manner with the drug sponsor. It has more of a peer review mechanism where instead of having government bureaucrats who are making decisions, you have real doctors who are taking care of patients that are leaders in their field who are part of the process. They're actually called rapporteurs, which kind of gives you the idea they're getting a rapport going with the drug sponsor, and you're trying to, in a problem-solving, give-and-take way, try to figure out how can we move this forward and figure out what its safety issues are, what its efficacy issues are, what's the right niche, the right kind of patient, the right kind of physician to be prescribing it. Let's try to find a path to success as opposed to a more obstructionist sort of approach that we tend to use in this country where, because of our conflict of interest laws, we don't allow peer review really in the process in any real way. And because frankly, there's also a lot of pressure, I think, on Congress and the FDA not to make mistakes. And so the specter of making a mistake, but one of the things that I think a lot of people talk about is getting the patient into the dialogue. That the risk-benefit equation for any new medicine is something the patient really ought to be part of the discussion around, and that's something that's also being lost. If you're a patient dying of cancer, what kind of risk are you willing to tolerate versus if you have hypertension or something like that? So it's a whole other discussion around this context, this conversation of the FDA reform. But it is something I think we should grapple with as a nation because a lot of those clinical development dollars are going overseas. We have great hospitals that could do the clinical trials, but they're going to Europe. And the European Union actually had whole commissions where they tried to figure out how can we be the best place to develop drugs, and that's where the money's being spent now. And we also should bear in mind that the FDA regulates goods and services that account for over 20% of the U.S. economy. So if the FDA is not working efficiently, guess what? Our economy is not working efficiently, and one of the most important things for future growth. Frankly, we should just take the F out of FDA, and get the D rather out of FDA and get, the drugs off in a different category, food and cosmetics and all that stuff separate. Make a more streamlined, efficient, elite organization that's really ready for the 21st century. I don't know what the possibilities are of actually doing that, but that's really, we need a rethink, and it's a new century, it's a time to really resolve this. It has great economic repercussions. If we can be the best in the world, all the companies are going to spend their dollars here. But right now, they're sending those dollars overseas and run their trials elsewhere.
Yeah, and it's in spades, and with devices, it's done.
Yeah.
And with drugs, it's going in that direction more and more.
Yeah.
And to your point about it, I had actually my last slide, which I didn't get to. I want to wrap this up and give you guys time for questions. It's this whole risk versus reward thing. We were getting our shoes shined ahead of time down in the, just so we can say we got our shoes shined. Not by a senator, but. And I was reading this article about, there's an ad about the number of teenage deaths from automobile accidents. And it's 4,000 a year in the U.S. Now, if that were a drug, it would never be sold. We would never let, if the FDA regulated cars,
there would be no
nobody
cars.
would buy it. But certainly not teenagers. But we have no problem with that risk versus reward ratio. But if a drug has the potential, and I'll give you an example, just to let me get on my soapbox a second for my last company, which has that drug in phase 3. This is for a cancer that I said, if you get this cancer today, you will be dead in 12 months. More than likely. You can get really lucky in the 2 years, but it'd be a really rough 2 years, lots of rounds of chemo, and then you're dead. So we got this drug that works. There's a companion diagnostic. You can say, do you have this genetic phenotype? And if you have it, then you take this drug. If you have it, it's going to, probably 50% chance that you will respond to this, and you'll have side effects like an aspirin. You'll take it every day, and you may live an extra year or 2 or 3. So that's kind of a no-brainer if you're the patient, right? But we saw a little bit of QT prolongation, which is a potential heart issue. And if you have this, there might be a 1 in 1,000 chance that you get a dysrhythmia and die of heart attack. Now, if you're a patient and you had this disease and your doctor told you that and you worked with your physician, don't you think you would take that risk, 1 in 1,000? Of course you would. But nonetheless, this trial is going to take a lot longer because they want to make sure that this is safe. And I used to have dreams when I was the CEO of this company that I was on a boat and I had a drowning person and I was going to throw them a life raft, and this guy from the FDA said, or a life preserver, he said, no, you can't throw that in the water because it has lead paint on it, and you can't save that person because we may pollute the river. Because they're so darn, I think, over-concerned. They're regulating us to death, I think, when it comes to drugs. I think a reasonable risk-reward ratio. The other experience I had as we were advancing this drug is I would get calls from parents and relatives of kids who are 12 and 13 who had this disease. Typically, it's very rare for them to have it, but it's a big country. And they would call up and say, look, we talked to our physician, we'd like to get our child on this drug. Well, we had no mechanism for getting that child on the drug because we as a company couldn't take the risk. And the FDA wouldn't let us because they didn't want to take the risk. And so here you have a drug that could help kids. And if Congress could come up with some kind of safe harbor where physicians and patients can make a decision and not risk the investment companies are putting into new drugs, and not risk physicians and hospitals, but still give these people tools. And we don't have that kind of regime right now. So, 50, 60 years ago, if we were developing a drug, certainly a physician could take a drug out of development 50 years ago and give it to a patient who they think. But today, in today's regulatory environment, you can't do it. So I just say no to these parents.
And we, the Connect has worked with the Hastings Center on Medical Ethics to put out a paper that actually puts it in an ethical context, because the question becomes, you have the regulator who sees the bad and the potential bad of everything, while you see the company who thinks it's the best thing since sliced bread. So neither of them are fully objective in what they're doing, but both of them are fighting and not in the room anywhere ever until the very end on an advisory capacity is the patient who's going to be receiving that drug and could have a better quality of life or whatever. And so the Hastings Center proposed, and we helped publish the paper, that potentially it could be an ethical issue to leave the patient out of that. And the proposal was that Congress and the FDA begin to think about, is there a way to bring the patient into that process earlier to give them that kind of option where if you're going to die in a year, and maybe you can risk a heart attack if you know this drug might give you another 2 or 3 years, not to mass produce it and give it to everyone in the whole world, but for those targeted disease populations, giving them more empowerment and autonomy in that development process so that they can make their own decisions as to their own quality of life and moving forward. So if you go to our website, connect.org, we have that paper on there. It's called Third Seat at the Table, talking about the patient being left out of the room during that development process. I know it's after 3. I know folks got to leave. So Dr. Reed and Mr. Salk will be here. Feel free to come back and chat with them, and thank you all for coming.